In order to understand the TCA cycle, an overview is needed first. One molecule of acetyl-CoA produces the products 1 ATP in the form of GTP, 3 NADH molecules, and 1 FADH2 molecules. The TCA cycle is also extremely crucial to cellular respiration and also helps produce most of the ATP generated in the electron transport chain. The first reaction of the TCA cycle involves acetyl-CoA and oxaloacetate with the enzyme citrate synthase to produce citrate. H2O is also used in the reaction in order to yield coenzyme A at the end of the reaction. For the second reaction of the TCA cycle, citrate along with the enzyme aconitate yields the intermediate cis-acetate. Acetate is then used again to produce the final product of this reaction, which is isocitrate. Water is used during both steps of this reaction to both hydrate and dehydrate the final molecule.
In reaction 3, you start with isocitrate and use the enzyme isocitrate dehydrogenase and the input of an NAD or NADP plus, and you yield NADPH or NADH and an extra hydride ion. And through this process, you come out the other side with alpha-ketoglutarate, and I went ahead and drew the intermediate structures in there as well. In reaction 4, you start with alpha-ketoglutarate and you yield succinyl-CoA through the enzyme alpha-ketoglutarate dehydrogenase complex, and you add in a coenzyme A, an NAD+, and you yield an NADH, as well as a carbon dioxide molecule.
In reaction 5, succinyl-CoA is converted to succinate by the enzyme succinyl-CoA synthetase. GDP plus PI is put into the reaction, and GTP is produced along with coenzyme A. In reaction 6, succinate is converted to fumarate via the enzyme succinate dehydrogenase. In this reaction, FAD is put in, and FADH2 is produced.
In reaction 7 of the citric acid cycle, fumarate is converted to a carbanion transition state before its conversion to L-malate. This is done by the stereospecific enzyme fumarase. Hydration occurs during this reaction with the addition of a hydroxyl group and hydrogen ion. This is a reversible reaction. In reaction 8, L-malate is converted to oxaloacetate. This is done by the enzyme L-malate dehydrogenase. NAD+ is consumed, yielding NADH and a hydrogen ion. Oxaloacetate is removed to form citrate, driving the reaction forward. This reaction is reversible.